The type 1 interferon or IFN1 response, specific to vertebrates, shares striking similarities with the intracellular pathogen response or IPR of the invertebrate Caenorhabditis elegans, suggesting an evolutionary connection. The IFN1 response provides defense against viruses and other intracellular pathogens. Similarly, the IPR is a protective response against viruses and intracellular pathogens like microsporidia that infect C. elegans. However, the origin and extent of this similarity between IFN1 and the IPR remains uncertain. Researchers from the University of California, San Diego have conducted a comprehensive review highlighting the similarities and differences between IPR and IFN1 responses, including their negative consequences on organism development. Rig eye-like receptors, or RLRs, present in both mammals and C. elegans, are central to antiviral defense. In mammals, RLRs trigger IFN gene transcription, and in C. elegans, RLRs, specifically DRH1, mediates induction of the IPR by viral infection. Furthermore, enzymes involved in nucleotide salvage metabolism also modulate host immune responses during viral infections. Mutations in these enzymes lead to heightened IFN1 responses in humans and IPR activation in C. elegans. Proteasome inactivation, which can be triggered by infections with intracellular pathogens, appear to trigger two separate responses. One, it elevates proteasome subunit genes through SKN1 in C. elegans and NRF1 in mammals, and two, it activates the IPR in C. elegans and IFN1 response in mammals. The IFN1 and IPR responses are transcriptionally regulated by IRF family transcription factors and ZIP1 respectively. Notably, however, less than half of IPR genes are regulated by ZIP1, so there is likely one or more other transcription factors yet to be identified that induce IPR genes. Interestingly, immune system activation can also affect organisms negatively by redirecting metabolism toward defense, potentially causing developmental delays, metabolic disorders, and premature aging. Although similarities exist between C. elegans IPR and mammalian IFN1 response regulation, further research is required to clarify their evolutionary origins. Studying immune responses across diverse host species offers valuable insights into the evolutionary development of pathogen defense mechanisms, enhancing our ability to advance disease treatment strategies.